Hi everyone, I'm Dr. Anisha, your microbiology faculty, and in today's session we will be having a rapid biochemical differentiation between three important gram-positive bacteria which are Staphylococcus, Streptococcus as well as Enterococcus. And for their differentiation we will be talking about Catalase test, Coagulase test, different types of hemolysis shown by these bacteria, Lancefield grouping, as well as finally the working algorithm which is going to help us narrow down the identification of these bacteria. So to begin with, let us first talk about the basic differences between these bacteria that we must be aware of. So firstly, when we are talking about Staphylococcus, we need to know the family to which it belongs to. So it belongs to Micrococcaceae family, and similarly we have Streptococcus and Enterococcus. Streptococcus belongs to family Streptococcaceae. And Enterococcus, which was previously grouped under Streptococcus only, has now been moved to a different group, a new group, that is Enterococcaceae. Right? So now, to differentiate them morphologically, all three of these are gram-positive cocci, whether these are arranged in clusters, chains or pairs. So firstly, to differentiate them on the basis of morphology, we have the very first image wherein are we able to appreciate these small cocci, these cocci which are arranged as bunches, or can we say these are arranged as grape-like clusters? So are we able to appreciate that? So these are gram-positive cocci arranged in grape-like clusters. And why is that particular type of arrangement present? It is because of that Staphylococci, they have multiple planes of divisions. Multiple planes of division. Right? Moving on to the next image. So in this image also we are very easily able to appreciate so many cocci which are present as chains. Right? So cocci which are present in chains, whether it is a small chain or big chain. So different bacteria can also, different streptococci can also be differentiated further on the basis of the number of cocci present in a chain. So what is the longest chain? It is Streptococcus salivarius. So cocci which are arranged in chains, whether long or short, are streptococci. And what is the reason behind this type of arrangement? It is because of the presence of a single line or single plane of multiplication. So multiplication occurs in one plane. And that is also the case with Enterococcus, which are present in pairs, and the plane of multiplication is also one. So Enterococci also have a specific morphological characterization or characteristic. So these are present in pair, but they have a special arrangement, spectacle-shaped arrangement. So these have these are spectacle-shaped bacteria which are arranged in pairs. Spectacle is an important buzzword which is used for Enterococcus morphology, observed under microscope, right? So this differentiates the images or the microscopic characterization of these particular gram-positive bacteria. So now moving on to the biochemical differentiation. So firstly, all three of these are gram-positive cocci, right? And we have certain tests that we already discussed, that we have Catalase and Coagulase and certain hemolysis shown by these bacteria. So the very first thing that we do, the very first test that we perform looking at the colony morphology, it is the Catalase test, right? So what exactly is a catalase? Catalase is nothing but an enzyme. It is an enzyme. So what would be the role of an enzyme? It would be used for some kind of breakage, right? So this particular enzyme in gram-positive cocci, if this enzyme is present in any gram-positive cocci, it is going to degrade hydrogen peroxide into water as well as oxygen. So this is the role of this particular enzyme, catalase enzyme, right? And this test is basically used to differentiate the members of family, firstly Micrococcaceae and Streptococcaceae, and of course we have Enterococcus also. So this is a test which is used to differentiate members of Streptococcaceae family as well as Micrococcaceae family, right? So what is the principle? So this particular enzyme, it breaks down hydrogen peroxide into water and oxygen, right? And this particular enzyme also has prothrombin-like activity. So hydrogen peroxide, it is an oxidative end product of aerobic carbohydrate metabolism. So it is nothing but an end product, and if it is allowed to accumulate, it is going to be lethal to the bacterial cell, right? So it is lethal to the bacterial cell, and that is why we do not have to allow this to accumulate. And the basic principle, as we've already said, that this hydrogen peroxide, since it is lethal, it has to be broken down into easier compounds like water and oxygen. So these become non-toxic thereafter. So this hydrogen peroxide has to be broken down. So now how are we going to perform this particular test? So onto a glass slide, we are going to place a small colony. We'll pick up a small colony, we'll touch the center of a colony and with the help of a wooden applicator stick, and we will place that colony onto a clean glass slide. And after this, we are going to add 1 drop of 3% hydrogen peroxide. And what are we going to look after adding 3% hydrogen peroxide? We will look for rapid and sustained effervescence. What is effervescence? It is bubbling. So in most cases, bubbling occurs immediately, right? So in order to consider this as a negative test, we will only wait for 20 to 30 seconds. So if in case bubbling occurs late, it is going to be a negative test, right? So it usually occurs between 6 to 10 seconds. So we are going to give it a positive test of rapid and sustained effervescence. And why are we not considering any positive result after 30 seconds? Because RBCs, if the colonies are present on the blood agar, so RBCs also have catalase activity. So we are going to misinterpret the result, and catalase, this hydrogen peroxide can be broken down by other compounds as well, right? So it is just the rapid as well as sustained bubbling. If it is seen immediately, then we consider it as a positive test. And how are we going to appreciate it? This is the effervescence we are talking about. Rapid and sustained bubbles after adding 3% H2O2, and this is a negative result, right? So now coming to the next important differentiation or next important test which is going to help us narrow down these bacteria is the hemolysis. Now hemolysis is also, since catalase is also an enzyme, so hemolysis is also caused by an enzyme which is hemolysin. And what do we understand by hemolysin? Hem stands for blood. And lysin, of course, naturally it comes down to lysis, breakdown of blood, right? So we have three types of hemolysis. One is alpha hemolysis. So can we write it down instead of 𝗲𝗵𝗃? Can we write it down as 𝗲𝗵𝗃? And if we write down 𝗲𝗵𝗃, so does it now make sense? 𝗲 and 𝗲. So what is 𝗲? Can we say that this is partial hemolysis? Partial hemolysis. So it is the partial type of hemolysis. Alpha hemolysis is not the complete breakdown of RBCs, but the membrane remains intact, wherein there is smaller amount of lysis of the RBCs, right? And similarly, we have other type of lysis, which is beta hemolysis. Beta hemolysis stands for complete hemolysis. Complete hemolysis, as well as gamma hemolysis is no hemolysis. This is complete breakdown of RBCs, complete lysis, and certain bacteria, they show no hemolysis on blood agar, checked on blood agar. So what is this blood agar comprised of? So into the nutrient agar, molten nutrient agar, we are going to add 5 to 10% of sheep blood at a temperature of 45 to 50 degrees. So this is how we prepare this blood agar, right? So it has 5 to 10% of sheep blood. So what are the examples of alpha hemolysis? Which bacteria show alpha hemolysis? So alpha males, they usually pretend to be villains. They pretend to be alpha, right? So it is the virulence streptococci, as well as Streptococcus pneumoniae, which show alpha hemolysis. So we are majorly covering up these gram-positive cocci. That is why our examples would be limited to these. So now coming to the beta hemolytic group. What are the bacteria which show beta hemolysis? See, beta hemolysis would be shown by Staphylococcus aureus. One thing, complete hemolysis. Then we can have group A, strep, which is Streptococcus pyogenes. as well as group pre strep, which is Streptococcus 𝗂𝗙𝗥𝗍. Right? So these are going to show complete hemolysis, whereas no hemolysis would be shown by Enterococcus. As already discussed, our discussion or this rapid differentiation is majorly confined to these bacteria, so we'll stick to them only. So now we have majorly understood the differences between these three hemolyses and the examples. So now let us understand this with the help of the image. So in the very first letter A, we have three types of hemolysis shown in a single Petri plate. So this is the blood agar, and we can very well appreciate three different patterns of hemolysis. So in this particular area, in the very first part, we have the bacterial colonies. These are the bacterial colonies, and around these bacterial colonies can we see complete clearing of the blood around the colonies. So are we able to see any discoloration? No. So this is the beta hemolysis. This is beta hemolysis or complete hemolysis. And in the next part, hemolysis although is there, hemolysis is present, but that is not complete, that is partial. And are we able to appreciate this greenish discoloration around the colonies? This is because of the compound or accumulation of 𝗲𝗏𝗡. 𝗲𝗏𝗡. So this greenish discoloration is present. So this type of hemolysis, it is partial hemolysis. Which is alpha. Alpha type of hemolysis or partial hemolysis. So what are the examples? It is the Streptococci viridans group and the Streptococcus pneumoniae. And in third part, we do not appreciate any kind of hemolysis around the colonies. No clearing, whether partial or complete, no clearing is seen around the colonies. So this is gamma hemolysis or no hemolysis at all. No hemolysis. Right? So now moving on to the next enzyme or the next test, which is the coagulase test. Coagulase, again, it is an enzyme. So it is an enzyme. And this coagulase has prothrombin-like activity. So what is the major role of this particular enzyme in staphylococcal infection? So this coagulase, it forms fibrin barrier. Suppose there's some form of infection, so it forms a boundary, a fibrin barrier around the infection. Now this fibrin barrier, it is going to help in localizing the wound. Localizing the wound. And what kind of wounds are usually associated with staphylococcal infections? Most of you must be knowing that we get carbuncles, furuncles, mostly known as boils. So these are confined. Small abscess of pus points are seen which are confined and they have a clear boundary around. They do not spread to the whole area, but they are completely localized. So that is the role of this particular enzyme in Staphylococcus infections, right? And what is the laboratory use of this particular test? So it is going to differentiate, or it is majorly used for identification of Staphylococcus aureus as well as Coagulase non-producing staphylococci, that are Coagulase negative Staph, right? So it is helping differentiate Staphylococcus aureus from cons, right? So now what are the two types of coagulase which are present? So we have one form which is bound coagulase, and the other one, it is the free coagulase. Now, both of these forms of coagulase, they have different activities. So, different tests are used. They have different enzymatic activities. So, different tests are used to perform or to test these particular enzymes. Two different tests, two types of tests are done to identify the presence of either bound coagulase or free coagulase. So bound coagulase, it is also known as clumping factor. It is also known as clumping factor. So one more important thing about this particular form of coagulase is that it is not present in culture filtrates, but it is bound to bacterial cell wall. Right? So remember, for testing bound coagulase or clumping factor, we are going to look for the presence of agglutination when this coagulase reacts with plasma having fibrinogen. So the particular end product of these coagulase or these factors or enzymes is the production of fibrin. It converts fibrinogen to fibrin, right? So now, to test this particular clumping factor or bound coagulase, remember that it is always done on a slide, right? So slide has so many edges. Can we say slide has boundaries? Bound coagulase test is done for something having sharp margins or boundaries. So a slide test is done for that. And so this free coagulase has thrombin-like activity. And this particular type of coagulase, it is present in culture filtrates. So this coagulase was attached to the bacterial cell, whereas this would be present in culture filtrates. So any coagulase containing bacteria or any bacteria that has coagulase activity is mixed with plasma in a tube, so a visible clot forms. A visible clot is formed inside the tube. This is because when coagulase, it reacts with a factor which is known as CRF or coagulase reacting factor. So now this coagulase reacting factor, which is a compound or a serum substance, it is finally leading to the formation of fibrin from fibrinogen. So it is the coagulase which is reacting with coagulase reacting factor which is present in the serum, which is leading to the formation of visible clot inside the test tube, right? So it is present in culture filtrates. So how are we going to perform these two tests, slide test as well as tube test? So onto a glass slide, for the slide test, we are checking for the clumping factor or bound coagulase. So onto a slide, we are going to make two circles with wax pencil, right? And onto one circle, we are going to have, in both the circles, we are going to have a drop of normal saline, right? And in the normal saline, we are going to prepare milky suspensions. And what are these milky suspensions comprised of? So it is the bacterial colonies that we are going to mix with the normal saline onto the glass slide, right? So why are we considering two circles, but we have to test only one of the test organism? So it is the control. Right? So suppose we have mixed multiple colonies into both of these circles, right? And now this control, it is a negative control, wherein a organism which is not having coagulase activity is going to be placed into this circle. And these are nothing but ATCC strains which you do not need to know about, right? So milky suspension of the colony has been prepared. Now after the formation, after complete emulsification of these suspensions, we are going to add plasma, a drop of plasma. And what is the role of plasma? This is going to help us appreciate the formation of visible clumps. These are chalky white clumps which would be appreciated, right? Whereas if we add plasma to this, since it doesn't have clumping factor, so milky suspension, no aggregates would be seen and the milky suspension will remain as is, right? So let us see this with the help of the image. So can you appreciate in this particular part of the image that we have milky suspension in the first section, in the first circle? So this is a negative test. And of course this is the control, wherein we have placed any such non-coagulase producing organism, right? And in this particular part or in this particular circle, are we able to appreciate these chalky white aggregates or agglutinates which are present? So this shows bound coagulase positive test. And how are we going to appreciate the formation of these aggregates? So after adding this particular plasma, we are going to rock the slide to and fro for around 2 minutes, right? And if there is no formation of clumps or aggregates after 2 to 3 minutes, then we are going to consider it a negative test, right? So it is very important to add plasma which is fresh, and we need to ensure that no older plasma is added, otherwise it might at times self-coagulate. So we have to ensure one thing for this particular test. So for a tube test, what are we going to do? Firstly, we'll have the bacterial suspension in the tube. We'll have the bacterial suspension, the bacteria mixed with normal saline, and to that we are going to add plasma. And after adding plasma, we are going to incubate this particular tube at 37 degrees for 4 hours in the incubator. 4 hours, right? So after 4 hours, we are going to look for clot formation inside the tube. Let's see that with the help of an image. So for this particular tube test also, it's ideal to take 4 tubes for one test. And what do these 4 tubes contain? One is a positive control, the other one is a negative control, the third thing is the test, and the fourth thing is the unseeded plasma. Why? Because we have to check that the plasma is fresh and there's no such self-clotting which can be present, which can occur in the plasma if it is not fresh, right? So one is going to be a positive control. That means that particular bacteria has tube coagulase or free coagulase, and it is going to show us a positive test of a visible clot formation. Negative control, wherein no clot would be seen. So these are also ATCC strains of the bacteria that we are going to. This is the particular bacteria that we are testing for the presence of free coagulase, right? So how are we going to read the result? So after 4 hours, we will slightly tilt the tubes and look for the flow of the fluid. So if it flows freely, so there is no clotting. But even if the suspension is flowing, we first have to look for the presence of even slightest of the clotting which can be present. Rest of the fluid might just move, but some clots might be present because there are certain degrees of clotting which can be appreciated. There's certain grades which you do not need to know about. So this is the clot, a very beautifully set clot, not moving even after tilting the tube. So this is a visible clot giving a positive tube or free coagulase test result, right? So for slide test, we have already discussed that after 2 to 3 minutes, we are going to, if no aggregates are formed, we are going to give it a negative test. Whereas for tube test, there is a slight variation. Since we have already incubated the tubes for 4 hours, and in case clot is not formed or it is not visible to you, we are not going to consider it a negative test, but we are further going to incubate it for 18 hours at room temperature. Very important, room temperature. Why not incubator? Because incubator might just dissolve the clot which might have been formed in 4 hours or 6 or 7 hours, right? So incubator might dissolve that particular clot. So it is 18 hours at room temperature further incubation which would be required to consider it a negative test. So after 18 hours, see, we will be reading the result the next day. So after 18 hours, if no visible clot is formed, then we consider it a negative test, whereas even if slightest clot is formed, we are going to give it a positive free coagulase test. Positive free coagulase test. And this is how we are going to interpret the results. So now moving on to the next section of this session is the Lancefield grouping. So Lancefield grouping is done for streptococcal species, right? So for coming down to this Lancefield grouping, we have already discussed that first thing that we are going to look for in any particular bacteria, gram-positive bacteria, is going to be catalase test, and the next thing is the hemolysis. So it is for Lancefield grouping, it is the beta hemolytic streptococci or streptococci which show complete hemolysis of RBC. They are further grouped into 20 serogroups A to V except I and J, and these groups are known as Lancefield groups, which are based on group specific 𝗕𝗵𝗃𝗏𝗏 antigen. So Lancefield grouping, it is based on group specific 𝗕𝗵𝗃𝗏𝗏 antigen. So it is just the beta hemolytic streptococci which are further speciated or grouped into Lancefield groups, and under these serogroups we have around 20 serogroups. Group A being the first, which is the Streptococcus pyogenes, which is further grouped or serotyped or genotyped. So Griffith typing based on M protein is done into more than 150 M serotypes, and genotyping, of course, it is going to be based on some gene, so it is M gene. So it is the M protein for Griffith typing and N gene for genotyping, which is grouped into more than 200 M genotypes. But for you to remember, the most important thing amongst this particular flowchart is the Lancefield grouping, which is based on group specific 𝗕𝗵𝗃𝗏𝗏 antigen. So we have 20 serogroups. So what are these Lancefield groups? Most important, group A, it is Streptococcus pyogenes. Then we have group B, which is Streptococcus 𝗤𝗙𝗙𝗃𝗏𝗄. So how do you remember group B? Because it gets confusing to remember these names. So I learned it with the help of a mnemonic BAMS. So it is group B, known as 𝗤𝗙𝗙𝗃𝗏𝗄, and it is responsible for meningitis and sepsis in babies. Right? And we have group C and group J, which have both of these include Streptococcus galaxy. Now, this galaxy has both group C and group G antigens, G and C antigens. And group C has, this includes Streptococcus equi. So these are all beta hemolytic streptococci. We are not considering alpha hemolysis or no hemolysis. These are all beta hemolytic streptococci, which would be further typed. These are typed into, or these are grouped by Lancefield grouping based on C carbohydrate antigens. So this particular classification is not just done by Lancefield grouping. A multi-top test is also now being used to serotype or to type these particular different types of streptococci. So it is very important to group these organisms. And then we have group D, group E, F, etc. So let us now discuss about the working algorithm. So firstly, we are looking at gram-positive purple-colored organisms. Purple-colored organisms. So can we say that these can be bacilli, cocci, and branching filaments? So since we are considering cocci, let us stick to cocci. So these are gram-positive cocci. So what is the first test that we are going to perform? It is the Catalase test. So this Catalase test can be a positive test and it can be a negative test. Positive catalase, it is given by Staphylococcus, whereas a negative catalase test is going to be given by either Streptococcus or Enterococcus. So this particular test, as we've already discussed, was used to differentiate members of family of Micrococcaceae and Streptococcaceae, right? So it can be Streptococcus or Entero. So let us write Streptococcus here. So now, for further speciation or identification for Staphylococcus, we have discussed another test, which is Coagulase test. Coagulase test, whether it is bound coagulase or free coagulase, right? So Coagulase test, it can be a positive test, it can also be a negative test. So Coagulase positive would be Staphylococcus, Staph aureus, right? And negative would be given by cons, right? So 𝗘𝗵𝗆 susceptibility would be further used for further identification of cons, right? So now moving to identification of Streptococcus. So till now we have done two tests. One is Catalase test, the other one is Coagulase test, right? So now, since we said first was Catalase test, and for a Catalase positive we have already narrowed it down to a Coagulase positive Staph aureus and cons. And for Streptococcus, for further identification, we will look for the third test, or it is not a test, it is a property shown by certain bacteria. So we will look for hemolysis. Now for hemolysis, we have already discussed that we have three types of hemolysis, right? So one is going to be partial hemolysis, alpha hemolysis. Now for further identification or for further speciation, we have already discussed that alpha hemolysis would be seen by Streptococcus pneumoniae and virulent streptococci. So what is the test that we are going to perform for further speciation? It is for alpha, can we say it has kind of similar pronunciation with O, AO. So Optochin susceptibility or sensitivity test would be done for further identification of alpha hemolytic streptococci, right? And the next one is beta hemolysis. So for beta hemolysis, for further identification and speciation, we will look for, with B, we have Bacitracin, susceptibility and PYR status. So this particular test is further helping differentiate the different types of beta hemolytic streptococci. And in third, we have gamma hemolysis or no hemolysis. For further identification of organisms or streptococci showing gamma hemolysis, we will be doing this 6.5 growth in 6.5% NaCl and PYR status. For further identification of whether it is Enterococcus or Streptococcus galolyticus, so we are going to look for 6.5% NaCl growth. And with Optochin susceptibility, we also have to look for bile solubility for alpha hemolytic streptococci. So this is how we will come down or narrow down to different speciation of the particular bacterial groups. So this is the whole working algorithm, and these were the important tests which were initial biochemical tests used for, or these are being used as initial steps in identification of bacteria. Thank you.